Hi, my name is Alex Sundermann, and my project for the SHEA Epi competition is entitled Enhanced Surveillance Methods for Genomic Epidemiology in Healthcare. Whole genome sequencing surveillance is an emerging method to sequence isolates regardless of the presence of an outbreak and often uncovers many outbreaks that would otherwise go undetected. When we investigate these outbreaks, we often see clues of potential routes of transmission, although lacking specific knowledge on how the pathogen was actually transmitted. Whole genome sequencing surveillance improves the detection of outbreaks, but the interventions remain unchanged. The questions here are if targeted environmental cultures can add to this and improve interventions, and also if the duration of pathogen shedding into the environment have any correlation with transmission. This study would be carried out at our hospital at UPMC Presbyterian. Currently, our hospital performs weekly real-time whole genome sequencing surveillance of major bacterial pathogens through an NIH-funded project called EDSHAT. We have sequenced over 2,000 bacterial infections in the past year and see new weekly outbreaks that we intervene upon. The objective of this project is to supplement the surveillance program with targeted environmental surveillance. Together, this would create an enhanced detection and precision of determining our transmission routes and thus enabling better IP interventions. Aim 1 of this project is to identify environmental reservoirs in the hospital and define their role in transmission. This hypothesis is that targeted environmental cultures will better identify transmission pathways. Here, targeted environmental surveillance means selecting high-touch areas within patient rooms or medical equipment which are suspected when we detect outbreaks. Our outcome would be the genetic relatedness of environmental isolates to that of patients. Aim 2 is to determine the impact of persistent pathogen carriage in infected patients on environmental contamination and transmission. Our hypothesis is that patients will persistently shed the same strain into their environment for a long time after their initial clinical infection. With this, we will measure patients that have persistent carriage who also appear to be a source of transmission to the environment and to other patients as well. To accomplish Aim 1, we will look at hospital locations or equipment that have implicated in weekly outbreaks. We will then perform targeted environmental cultures, sequence them, and compare with our patient isolates and other environmental isolates. For Aim 2, we will follow patients in Aim 1 who have shed related isolates into their environment and perform follow-up environmental cultures throughout their hospital stay and readmissions. This is a diagram depicting the methods of the aims. We have an initial cluster detection of 2 patients and will perform the targeted environmental cultures. We will look for an identical environmental isolate and continue to monitor for additional patients within the cluster and any environmental commonalities. For the patient with a matched environmental isolate, we'll perform their environmental cultures in Aim 2 by performing systematic targeted environmental cultures over the course of their stay and readmission. In Aim 1, we will look for trends of which surfaces or equipment are most often contaminated that are related to patient isolates. We will also look for any temporal trends in environmental isolates to future patient isolates as possible sources of transmission. For Aim 2, we will look at the distribution of environmental contamination and factors that may contribute to prolonged shedding or transmission to other patients. We have some preliminary data to suggest persistent carriage of related isolates. Looking at our genomic surveillance data of patient isolates over 3 years, we see prolonged carriage of the same isolate of which had evidence transmission to other patients. The figure here depicts an example of a patient with Klebsiella pneumonia infection with related isolates 373 days apart and possibly transmitted to another patient on a shared unit 220 days after the initial isolation of the first culture. We also have preliminary data showing how surveillance data can be linked to environmental culturing. We previously described an outbreak of 6 patients with Pseudomonas aeruginosa infections, where all but one exposed to a common gastroscope. That gastroscope was subsequently culture positive for a genetically related Pseudomonas aeruginosa isolate, implicating it as a transmission route. The $20,000 requested would be dedicated to supplies for environmental cultures and sequencing. The data generated from this project will help aid infection prevention interventions by enhancing the precision of interventions, as well as to better understand transition dynamics within the healthcare setting. This data will also be applied to a future NIH training grant. I have a stellar mentorship team here at the University of Pittsburgh with Dr. Lee Harrison, who leads our Center for Genomic Epidemiology, Dr. Graham Snyder, who directs the UPMC system-wide IP program, and Dr. Daria Van Tine, who is an expert in microbial genomics. Thank you for listening, and see everyone in Seattle this April.